Hello, everyone, and thank you for attending today's webinar, Driving Quantitative Image Analysis for Early Phase Clinical Trials with Metadata and MedPace, presented by Metadata and MedPace. I'm Michael Shoshua, and I'll be moderating this webinar. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there's no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Now let's begin. Scott Holland of MedPace will introduce the speakers. Go ahead, Scott.
Hi, I'm Scott Holland. I'm the director of the Imaging Core Lab at MedPace. And I'll be taking us through the program this morning with some brief introductions of our subject matter experts who are going to be speaking on the specific topics. First, I'll give a brief overview of quantitative imaging biomarkers for clinical trials, and then we'll focus on some examples with our speakers in oncology, neurology, and metabolic diseases. And hopefully, we'll have a little bit of time at the end for some questions and answers. So let me begin with Andrea Falkoff, who is the Director of Clinical Imaging Delivery at Metadata, and of course myself, and then Lion Gleish, who is Vice President of the Medical Department here at MedPace, specializing in hematology and oncology trials, Dr. Richard Shire, Vice President also in the Medical Department, specializing in clinical trials in neurology and pharmacology, and lastly, Piotr Kreski, who is also a Vice President in our Medical Department, specializing in trials in gastroenterology and hepatology. So a little bit about the Metadata-MedPace partnership, which is really the focus of this webinar. MedPace Core Labs brings unique capabilities to the partnership with Metadata, with more than a dozen MD and PhD level imaging scientists from backgrounds in radiology, cardiology, medical physics, and image processing, and access to more than 50 full-time physicians across therapeutic specialties at MedPace. Our Imaging Core Lab staff manage imaging endpoints for clinical trials across all medical imaging modalities and around the globe. Our advanced image quantitation services make us the ideal partner for small to mid-sized biotech companies worldwide. MedPace Core Labs has dedicated quantitative image analysis software development team based in our office in Lyon, France, with the capacity to construct customized quantitative image analysis pipelines that can plug into Rave Imaging to provide the imaging biomarkers required for any trial. This next slide illustrates the integration of Rave Imaging and MedPace Core Labs quantitative analysis pipelines. At the juncture of these two circles, you see imaging data integration. The sites begin with uploading images into Rave Imaging, and Rave Imaging already provides image management support for a wide range of clinical trial marketplace, providing electronic image capture for clinical trials, integration with MedPace Core Labs validated quantitative image analysis pipeline, adds inline quantitative analysis functionality capabilities with support from MCL imaging scientists with expertise across the entire spectrum of medical imaging, from CT, MRI-based resist measurements to liver fat fraction measurements to echocardiography and beyond. Clinical sites benefit from the support they receive from MedPace Core Lab imaging experts while experiencing the convenience of Metadata Rave Imaging for image data uploads and tracking. Sponsors benefit from the familiarity and reliability of the Rave Imaging interface. The integrated pipeline can now be used for defining eligibility, for monitoring disease progression or response, for making no-go decisions, and for providing quantitative endpoints. So with that, I'll hand the microphone over to my colleague, Andrea Falkoff, to talk a little bit about Rave Imaging.
Great, thanks, Scott. I'm going to start by reviewing the basic functionality of our Rave Imaging product. The tool is extremely flexible and can support any type of imaging trial, whether it's simple or complex. One feature that I want to emphasize is that the system is web-based, which means it does not require users to download or install any software. Rave Imaging can be used to collect and de-identify images and other data from sites, and then move those images to a QC step that is completed by MedPace's project coordinators and techs. Once this critical QC step is performed by the MedPace team, the images are automatically moved to an independent review step, where MedPace's central reviewers perform their quantitative analysis. Forms can be configured as well for the purposes of documenting image QC or results of an image analysis. All forms can be configured with edit checks as well as info bubbles that provide standard guidance to the readers right on the form. Rave Imaging also takes the results of various reviews completed by MedPace and can generate a safety or eligibility result sheet that sites can access all within the same platform, or those results can even be presented within Rave EDC. Because the system is so configurable, it can handle any type of image or data and support any type of image analysis across all therapeutic areas. This workflow really illustrates the synergy that comes from partnering Rave Imaging with MedPace's expertise and processes. Rave Imaging provides great value to site users as well. It allows for single sign-on through Rave EDC, so sites don't have to remember multiple logins. The system is intuitive and user-friendly, and it has features like edit checks that ensure the right images are being uploaded at each visit. This really helps streamline the process for sites, ultimately reducing errors. On an oncology trial, for example, we can configure the system to require a CT scan, and if for some reason the site can't provide this, a comment is automatically required, reducing the need for queries later on. The system also has automated de-identification of DICOM tags to remove protected health information and access to pixel masking tools to remove PHI when it's burned into the image. In addition, when integrated with Medidata's Rave EDC platform, it minimizes duplicate and manual data entry of subject and visit information by site. For example, if patient weight and tracer information are required for a PET analysis, this information can be automatically pulled into our imaging platform from Rave EDC, saving the site an extra step. All these features are available to site users without the need for any software installation and is compatible with all modern browsers. In fact, in a recent survey of site users, we found that sites prefer our system 3 times more than any other imaging software tool. This really speaks to the ease of use of the system and the available features for site users. So the Medidata platform also has very flexible workflow management capabilities that can be easily pre-configured to support all of MedPace's trials. For example, once data is uploaded by a site, that data can be automatically routed within the system to a quality control step where a MedPace tech can complete and document image QC. If quality control fails, a query can be automatically generated and issued to the site. If quality control passes, the images can be automatically routed for quantitative analysis by MedPace's read team. Several workflows can be configured simultaneously as well, and the system also automates adjudication triggers and presentation of data to other users. For example, on a melanoma trial, you may need to collect CT scans, photos, and clinical data. All this data can be easily uploaded onto the system and routed to the appropriate physicians. You may want to send photos to the dermatologist, the images to radiologists, and then route the dermatology and radiology results, as well as the clinical data, to an oncologist for a final assessment. The system can seamlessly handle routing the right data to the right reader at the right time. Medidata's strong imaging platform, combined with MedPace's extensive experience with quantitative imaging biomarkers, really allows our partnership to deliver high-quality results for your trial. In summary, Rave Imaging brings values to all stakeholders. It ensures the correct data required at each visit is included in the submission, ensures that images are de-identified, and then sends those images through MedPace's pre-specified workflow in an automated fashion. Our technology, combined with MedPace's ability to deliver high-quality results, allows us to support all types of quantitative imaging biomarkers. This automation reduces the burden on sites and the MedPace team, reduces query rates, and helps increase the quality and turnaround of image submissions, all things that are very important for clinical trials. When images are submitted, all activities are also visible within the system in real time through the user interface, but also available through reports within the system. All these features combined help reduce costs, timelines, and improve data quality, regardless of the quantitative biomarker being assessed for your trials. I'm now going to turn it over back to Scott and the team to speak more about MedPace's experience in this space.
This is Scott from MedPace, taking the microphone back. So I'm going to give a little overview of quantitative imaging analysis biomarkers. So the slide you should be seeing now is describing a series of 6 FDA guidance documents on endpoints for clinical trials that involve imaging. These are all published in the last 3 years, from 2018 until present tense. Clinical trial endpoints for approval of cancer drugs, clinical trial imaging endpoints, process standards. There are 2 documents on liver disease with non-alcoholic steatohepatitis with fibrosis, and also non-alcoholic steatohepatitis with compensated cirrhosis. And finally, technical performance assessment on quantitative imaging in device pre-market submissions. There are also a number of documents on digital image data formats and storage. So all of these are relevant to the role of a core lab and Rave Imaging in clinical trials. MedPace Core Lab simplifies the complex image analysis that happens behind the scenes in generating quantitative imaging biomarkers, and the seamless integration with Rave Imaging workflows allows streamlined image management and facilitates expedited go, no-go decisions about eligibility and trial continuation. So I'm going to go through a few examples of the integration of Rave Imaging workflow with MedPace Core Lab's quantitative analysis pipelines. MedPace physician subject matter experts will demonstrate the power of the partnership we've established and its utility for site investigators and independent central reviewers for assessing patient eligibility and disease progression in 3 distinct therapeutic areas for clinical trials. These include oncology and metabolic diseases, as well as neuroscience clinical trials. In each case, we'll show the ease with which imaging biomarkers can be generated and fed directly into Rave Imaging ECRS and ultimately into Rave Imaging EDC. MCL quantitative analysis pipelines all incorporate key features that facilitate image display and quantitative analysis at the site or in the hands of skilled technologists or central reviewers. These universal tools are available across quantitative analysis applications and include a DICOM viewer, ROI tools, auto-population of Rave Imaging ECRF with the results of measurements, in some cases auto-calculation of responses, for example, oncology response criteria, and automatic reporting of eligibility. These capabilities then seamlessly integrate with Rave Imaging and Rave EDC, and they're fully validated. And I should also mention that all of this capability is 21 CFR Part 11 compliant with an audit trail for each step. Systems are also SHIELD certified for studies that are taking place in the EU. We have data security in place for those trials as well. So with that overall introduction, I want to introduce Lane Gleisch as our first subject matter expert to talk about oncology biomarkers. So Dr. Gleisch.
Thanks, Scott. So obviously imaging is an important part of oncology trials, especially if response or PFS is being used as an endpoint. Laid out on this slide are a lot of the different imaging modalities that our imaging core lab has experience with, and obviously they can work in modalities beyond this as well. Things are a little more straightforward for RECIST 1.1 than for everything else because RECIST 1.1's been around for quite a while. The site reads have utility there, but even still it's worth collecting the images and considering doing central imaging with a RECIST 1.1 study, just in case issues come up with the FDA, whether on an early phase study or a late phase study. If you don't collect the images prospectively, it's very difficult to get them in later on. And the Rave MedPace combination provides a very nice way to get the images uploaded We do actually happen to also like the Rave metadata system for the EDC and the ability to manage the investigator reads also and to compare final analysis of investigator versus central reading as well. Central reading becomes more important for other systems, especially RANO, for example, or PERSIST, where there's going to be more variance. They're not as commonly used. You're not dealing with just a single measurement. And the use of the additional technology that's been added into our system where the reader can circle the lesion and the system helps measure the tumor size so that you get more consistency between the readers as well has advantages. This can also be applied at the site level if the site is comfortable using that system as well, so the site readers can use this system that's been developed as well. And it's also able to auto-calculate the sizes and have that then get entered into the central read. So it's a nice integration of the EDC with the central imaging from a site perspective as well. If they're already doing the study on Medidata Rave for the EDC, to be able to upload the images through Medidata Rave is an advantage. And for operationally to be able to track the receipt of the images by a combination of the central imaging lab along with our trial managers is also a great advantage, because often one of the biggest hurdles is actually getting all the images in and getting complete images in and being sure that's all been received. And then you can also utilize the clinical data portion of Rave based on what's put into the imaging charter for what clinical data will be utilized for decision making as well by the central readers. So all in all, a good system to have benefits in terms of the central reading. I'm going to turn things back over to Scott for some image examples, and we'll take it from there. Thanks, Scott.
Thanks very much, Dr. Gleisch. So I just want to kind of give a demonstration of what one of these quantitative pipelines looks like for oncology. So this is an example showing tumor measurements in the liver. And you see here at baseline, we have 2 target lesions. We've used the ROI tool to identify them and outline them, and then the orthogonal longest diameters are automatically generated and fed back into the EDC. And then at the next time point, we see follow-up imaging, where these 2 target lesions are again identified, and their longest diameters are fed into the EDC, but now we also see there's a non-target lesion that has appeared in the imaging at the follow-up time point. And so the tumor progression is calculated automatically based on the target lesion diameters. This would be a partial response. Based on the appearance of a new target lesion, this becomes progressive disease. So the overall response is fed back into the Rave ECRF as shown above. Hopefully that's large enough print that you can read it. And then on to the next slide. This demonstrates some of the built-in edit checks that we have in our quantitative analysis pipeline. So for example, in this case, the central reader attempts to identify a 3rd target lesion at the follow-up time point, but this target lesion is also in the liver. And as you know from the RECIST 1.1 rules, you're only allowed to have 2 target lesions per organ. And so the a little dialog box pops up and says, you know, you're not allowed to do this. And so the error has to be corrected before the ECRF will be populated. So that gives an overview of our quantitative analysis capabilities for oncology. These modules can be, as I mentioned, we have a software development group that can modify and customize these for any particular disease criteria that you're interested in. So if you have an immunoresponse therapy for brain, that can easily be set up in one of these quantitative pipelines and data fed back into Rave Imaging. In the next example, we're going to talk about some of the applications of neurology imaging biomarkers, and I'll hand the microphone over to Dr. Richard Shire for that.
All right, thanks, Scott. In neuroscience, various CSF and blood-based biomarkers have been used to confirm both target engagement and the impact of treatment on disease. But these do not provide information on specific brain regions, and some markers, such as circulating A-beta, may be largely of peripheral rather than brain origin. Imaging may be used to assess safety, such as hemorrhagic transformation in stroke or amyloid-related imaging abnormalities in Alzheimer's, but most interesting is the use of quantitative assessments as predictors of disease progression and as markers of treatment effect. The use of MRI in counting new or enhancing lesions in multiple sclerosis is well established. Imaging is also demonstrating utility in a range of neurodegenerative and neurodevelopmental indications, and we're seeing increased use also in muscle disease. Imaging has roles in all phases of development, but the greatest importance of reproducible quantitative imaging in neuroscience has been in phase 2 proof of concept studies. MedPace imaging scientists are involved in clinical studies beginning with consultation on the optimal design and practical considerations for imaging protocols, working to balance the information yield with the imaging time and with other factors impacting site and subject burden. The quantification of images includes automated measurements of whole brain and of specific cortical and subcortical structures, depending on the therapeutic indication and sometimes upon the mechanism of action of the therapy. With assistance from the MedPace core laboratory and using Rave Imaging, quantitative brain mapping becomes practical for multi-site clinical trials. The core laboratory pipeline automatically segments and measures volume of more than a dozen brain areas, avoiding the subjectivity associated with manual outlining. Still, the accuracy of the automated systems cannot be taken for granted, especially in areas of pathology, and expert neuroradiologists inspect the results of the automated processing and make corrections as needed. For studies in which changes in brain volume over the course of disease or as a function of treatment is the endpoint of interest, simple subtraction of the brain volumes between time points leads to noise due to differences in segmentation and image normalization. The boundary shift integral assesses changes in segmentation boundaries over time, and the KN-BSI method has been shown to provide the greatest accuracy in longitudinal brain volume change estimation. Core Laboratory has implemented and validated this method for studies where longitudinal changes in regional or home brain volumes are needed. The results are automatically fed into Rave Imaging, streamlining the process and avoiding the potential for transcription error. In the next segment on metabolic imaging endpoints, you'll hear from my colleague Piotr Krecki about the MRI proton density fat fraction method. So I won't go into the details here, but we'll share an example from the vastus lateralis and other thigh muscles in a pediatric patient in a trial of a treatment for Duchenne's muscular dystrophy. The MedPace Core Lab has developed a pipeline for muscular dystrophy trials, quantifying total and contractile muscle volume to measure fat fraction, as well as T2 as a marker of muscle injury. We've implemented this approach in both single-site and global studies, and we'll be presenting data on the reproducibility of these measurements at the World Muscle Society meeting later this year. The Core Laboratory analysis pipeline computes the parameters and then passes the data back to the Rave Imaging ECRF. And the key to success for all imaging analyses has been streamlining the process, maximizing reproducibility, and making life as simple as possible for the sites, the readers, and the data managers. And with that, I'd like to pass things over to my colleague Piotr Krecki, who will discuss metabolic imaging biomarkers.
Thank you, Rick. So moving from oncology and neurology to application of MR imaging in the field of metabolic disease. Here, one of the research focus areas that has gained significant thrust in the last decade or so has been non-alcoholic fatty liver disease and steatohepatitis or NASH. In simple pathophysiological terms, NASH is characterized by excess liver fat accumulation with ensuing lipotoxicity, with liver inflammation and fibrosis. I would like to talk about one of the MR-based modalities of liver fat assessment, the proton density fat fraction, or MRI PDFF. Being non-invasive and standard, acquisition using a multi-echo gradient echo sequence available on most MRI scanners, it has practically replaced other imaging techniques for assessment of liver fat content in quantitative research setting. Actually, some researchers believe that PDFF is more reliable than histology itself for liver status assessment because it covers a larger amount of liver tissue than a histological biopsy, and unlike the widely used histology scores such as the steatosis component of NASH CRN's NAFLD activity score, it provides a truly quantitative assessment of liver fat in a wide range of clinically significant liver fat content. As many in the audience will appreciate, the use of non-invasive biomarkers is highly encouraged by the regulators and promulgated by the FDA and EMA guidance documents for drug development in NASH, and the utilization of PDFF in NASH research fits perfectly into this regulatory recommendation. In terms of groundwork technicalities, in our trials, med-based core lab staff work with sites to implement and qualify a 6-echo sequence that allows for separation of fat and water signals with corrections for relaxation time of different lipid peaks. The acquired multi-echo data is then analyzed using Medbase's proprietary MRI PDFF quantitation pipeline based on the well-validated methodology. The MRI PDFF quantitation pipeline yields liver fat maps, as shown on this slide here, for different steatosis grades on the histology-based semi-quantitative steatosis grading scale. Average liver fat fraction can be calculated from these maps in a number of different ways. Typically, 3 regions of interest or ROIs are selected, 2 from the right lobe and 1 from the left lobe, and the average of the 3 ROIs is calculated as the proton density fat fraction percentage. The ROI selection is performed by technologists at Medbase and then verified by a central radiologist. We found this method very efficient. It provides a robust estimate of PDFF and agrees with more consuming methods. As you can appreciate on this slide, the interclass correlation coefficient lies somewhere between 0.99 and a unity for different combinations of different numbers of ROIs from 3 to 9. Actually, this research was done at Medbase by Scott and the MCL group, and found that 3 ROIs that are widely dispersed across the 2 liver lobes give a very accurate estimate of the average fat fraction in the entire liver. Here, I just wanted to briefly demonstrate the flow of data processing. Qualified sites upload imaging data to Rave Imaging. The Medbase core lab perform the QC, file queries as needed to sites, and accept the final MRI scans from sites. This process is done in ClinTrack imaging. The MRI PDFF pipeline is run by technologists, and finally the PDFF results are approved by the central radiologist and entered into the Rave Imaging ECRF for that specific time point. All this ultimately is leading to study outcome data. On this slide here, you can see a sample of published PDFF results from a number of NAFLD trials, relatively recent. As you can see, whether looking at mean relative or absolute change from baseline on the top two panels, or a proportion of PDFF responders defined as a certain minimum threshold on the bottom panel. The MRI PDFF shows pretty nice responsiveness with compounds from different classes, be it FGF21, FXR, agonists, ACC inhibitor, or thyroid hormone agonists. PDFF differentiates significantly from placebo, and this is in relatively short time frames, ranging in these examples here from 12 to 16 weeks, and relatively small sample sizes as well, consistent with early proof of concept trials. With this, I would like to hand over to Scott for the closing remarks.
Great, thank you, Piotr, and the other speakers as well. Very nice coverage of some of our quantitative imaging biomarkers and their use in clinical trials. Hopefully that conveys the importance and relevance of adding these capabilities to a trial, and the ease with which that can be done in conjunction with the use of Rave Imaging, which is really widely used in the industry. So I just want to kind of reiterate a few points. The use of quantitative imaging endpoints in early phase clinical trials are really encouraged by FDA, and some of their guidance documents encourage further research in this area. And the rationale for that is these imaging biomarkers are sensitive to disease progression, often in a time frame that is more reasonable for conducting a clinical trial. For example, in oncology, we can't necessarily wait for clinical outcomes to demonstrate efficacy, whereas imaging outcomes may reflect change in disease status earlier than clinical outcomes. These types of clinical or quantitative imaging biomarkers have been validated in key therapeutic areas, so we can have confidence in using them. And they are easily implemented in this partnership between Medidata and MedPace, making them readily available to sponsors and investigators for use in the clinical trial environment. I also want to just sort of point out that the use of imaging really does provide a cost-effective solution for efficacy and safety endpoints in early phase clinical trials. As I mentioned, often this data can be collected earlier than clinical outcome data, and often at a lower overall cost. So I think that wraps up the formal presentation component of this webinar. And at this stage, we'll move into answering some questions that are coming in online. I see that we have a couple of questions already, and I will sort of filter these and direct them to the appropriate speaker. The first question is on the neurology segment. Dr. Shire mentioned that radiologists validate the results and can make corrections, and the question is, how are these corrections used to enrich the model? And I'm assuming that means how are these corrections used to provide a more accurate result of volumetric measurements? So I'll, as the author of some of these quantitative pipelines, I'll take a first shot at that, and then I'll pass the microphone over to Dr. Shire to comment further. So when quantitative analysis is run in an automated fashion, we generate a visualization of the outlines of key brain structures. For example, whole cerebrum, whole cerebellum, gray matter in cerebrum, white matter in cerebrum. We can also generate volumes of some of the areas that were shown in the slides in deep brain gray matter nuclei, such as caudate, putamen, lobus pallidus, thalamus, hippocampus, and other structures. But, as is often the case with imaging data, there is a good deal of variability in terms of both the quality of the incoming data and the contrast and signal noise that occurs in these images. And as a consequence, the automated methods sometimes run into problems. So it's really important to have someone inspecting those results on a case-by-case basis. So initially that's done by the technologist, but no one has the knowledge of cerebral anatomy in brain imaging to the extent that a neuroradiologist would do. And so the final stage of approving these boundaries and outlines is done by a neuroradiologist, and that gives us some confidence that the results are accurate, and we also can sign off on that as a result that's been passed on to clinical trial submission. So Rick, do you have any further comments on that?
Yes, so thank you. When we're looking at the boundaries, we're thinking about what applies to an individual image or the images for an individual subject, and there, Scott's nicely covered it. We also have the consideration of how we can help our automated processes do a better job in the future, and that's typically something that's looked at as the algorithms are refined from study to study, but for a given study, we try to keep things locked down so that we have maximum consistency.
Great, thanks a lot. We have another question which I'm going to read and hopefully ask Dr. Gleisch to comment on. First of all, a compliment on a very nice presentation, but asking us what do we think is the most challenging regulatory step with submissions that include quantitative imaging endpoints. Lion, do you have any comments on that? You're very familiar with it.
Yeah, you have to look at it from the FDA point of view, and speaking strictly oncologically here, and Pazdor's commented on this multiple times during his decades as head of the oncology division, oncology office. Their preference is survival data. Data death has an exact date. Once we're using imaging, the date is controlled by the frequency of the image and the date of the image rather than the actual occurrence of when disease progressed or the patient had PFS. So with that as an understanding, the FDA will always have a degree of skepticism with any imaging endpoint because it's, in their point of view, never ever as good as survival data. Survival data in diseases that aren't like ovarian cancer or pancreatic cancer, where death is unfortunately quick occurrence. In other diseases like breast cancer or prostate cancer, it might be many years, and that's financially toxic to the pharmaceutical company developing the drug. So you have to rely practically then on imaging. The more you're relying on RECIST, the more that's acceptable to the FDA because that's been used repeatedly and they have confidence in RECIST. But even then, their confidence in the endpoints varies by tumor type. And in some of the tumor types, they'll accept PFS. In some, they won't. In some of the tumor types, they'll consider accepting, particularly for accelerated approval, response criteria. In others, they don't see it as a meaningful surrogate endpoint, and it's not acceptable. With that, then, when you're getting more novel in the system that's utilized, once it's not RECIST 1.1, even if it's iRECIST, for example, which is a published and respected system, the FDA has commented that they think it should be a secondary endpoint rather than the primary endpoint, that the RECIST should be the primary endpoint because it's a more tested system. When you're into the leukemias and lymphomas, you're in a different set of systems which are tested and are accepted, but the imaging is less crucial there typically. So I hope that answers the question. The improvements in the ability to make the reads more precise, more quantitative, obviously bolster the data, but still would never put it at the level of survival data. So with all that as an understanding, it's sort of a way to try to keep de-risking the program.
As you were answering that question, another one came in, which I think you also answered, basically asking if you have any oncology-specific examples for FDA-approved quantitative imaging biomarkers or any that are optimistic. And I think you mentioned RECIST 1.1 as...
Yeah, and let me if you try to go for something that's outside RECIST 1.1, there are not many. One that comes to mind whenever I talk about this is the ruxolitinib approval. The ruxolitinib approval the FDA has talked on and is very proud actually of having done it, because they approved ruxolitinib off of volumetric MRI, which the FDA has been upfront was what the sponsor wanted to use as their solo primary endpoint, and because it was novel, the FDA would not accept it as a solo primary endpoint. And they had the sponsor develop a patient-reported outcomes questionnaire as a co-primary endpoint. And they also had the sponsor send them additional data ahead of time about their volumetric data for their review before the phase 3 study was done. So the FDA is that cautious with a new and novel endpoint, and that's a very unique endpoint in terms of the volumetric measurement of the spleen, and that's the best example. If you look at the immunologic drugs that have gotten approved, the PD-1s, many of them used modifications of RECIST within their studies. Some called them different things. This was all prior to the development of iRECIST. Some called it IR-RECIST, some called it modified RECIST, but nearly all of them had a RECIST analysis in it. And if they didn't, I strongly believe that the FDA performed a RECIST analysis themselves from the data. Most were approved off PFS, where if they were using a minor alternate analysis, it wouldn't have affected it much anyway. Some were approved off survival. Only in limited tumor types were they approved off of response data. So PFS was the driver there, which therefore was much more driven by simple RECIST data.
We have a question related to the liver PDFF presentation. Basically asking whether MedPace offers multiparametric MRI to evaluate neuroinflammatory activity in disease, and what biomarkers are we using, and then also the second part of that question as to whether there's evidence that PDFF can predict outcome. So I'll take I'll make a quick comment on this from the quantitative analysis point of view, and then pass the microphone over to Dr. Kresge. MedPace Core Labs does use various imaging biomarkers, including as we are doing the PDFF mapping, we generate T2 maps that are then used to correct for the relaxation time of the various lipid peaks in the fat fraction maps. There's also been pretty strong literature showing that T1 is directly related to liver inflammation, which is mediated by water content in liver parenchyma. So we do have the capability of generating any of these quantitative maps from MR data. And unfortunately, there isn't a lot of peer-reviewed literature really directly endorsing the use of multiparametric MR methods as fibroinflammatory markers. There are certainly techniques that are out there that can be used in trials. Their admissibility and their validation in the peer-reviewed literature is still a little bit lacking. And then, Dr. Kresge, do you have any further comments on PDFF as a predictor of outcomes or fibroinflammatory markers?
Yes, thanks, Scott. With regard to the outcomes, this is a great question. Obviously, PDFF is the imaging biomarker of liver steatosis, and so the question is really down to whether liver steatosis is predictive of clinical outcomes. In some theories and pathophysiology of NASH, it is liver fat is associated with lipotoxicity, as I mentioned, and this leads further into inflammation and fibrosis. However, we don't know which patients will actually progress, advance into this inflammatory and fibrotic phenotype, and we know that only a minority of patients actually with excess liver fat will actually advance to that stage and further complications of NASH. So even though it is used oftentimes as a predictor in clinical drug development and it leads on to further trials, it is not considered as a surrogate marker. And as you can read in the regulatory guidance documents, we are reliant on other modalities, especially histology and inflammation assessed by histology and fibrosis assessed by histology for the prediction of clinical outcomes.
Great, thanks so much for those comments. There's a question that's come in for Andrea Falkoff from Medidata. And Andrea, the question is, does Rave Imaging support other types of data like documents, photos, or videos?
Yes, absolutely. So the system can be configured to accept pretty much any DICOM or non-DICOM data, including videos or PDF files. And in addition, for those of you who might have missed the audio in my section, just a few other features. Sites can use the platform using any modern browser. They actually don't need any software to upload any DICOM or non-DICOM data. And site users in a recent survey actually said they prefer our system 3 times more than any other imaging software tool. So this just speaks to the fact that the system is very easy to use for site users.
And then there's a follow-up question as to whether you're able to post imaging status information back to Rave EDC for the purposes of reconciliation.
Absolutely. So there's several use cases for integrating with Rave EDC. So we can certainly pull imaging status information into Rave EDC directly so that data managers or other team members can use them for reconciliation. And then a few other use cases, just to reiterate that I mentioned during my presentation, was we can also pull any data from Rave EDC into Rave Imaging. So if information is required by the site to include with the imaging submission, if it's in Rave EDC, we can actually automatically pull that over to save the user an additional step. And then if there's any information that's needed for any of the analyses that MedPace is performing, like any kind of clinical information or prior radiotherapy, for example, we can also pull that information automatically from Rave EDC.
Great. We have an additional question that has come in online, which is, what do you think is necessary to grow beyond novel endpoints being considered as just a secondary endpoint? Is it just experience or more trials? And Dr. Shire, would you like to make a comment on that?
Yes, there's a couple of pieces. The first is that the utility of an imaging endpoint, even if it's not definitive, may be that it can be used for decision-making in an early phase or mid-phase trial. It provides de-risking, provides confidence for a sponsor to take the drug to the next phase. So that's not necessarily going to serve as a regulatory approval, but still it's value added. There is also the potential, and we've seen this in some cases, where the marker can serve as supportive evidence for the accelerated approval to be confirmed by additional clinical studies with more clinically oriented endpoints. And then there's a process for qualification of a marker for actually serving to replace the clinical endpoint. And this usually begins with experience in natural history. For example, in the Alzheimer's area, the ADNI Consortium has provided valuable input on both volumetric endpoints with MRI and then also with metabolic endpoints with PET. And then ultimately for an actual primary approval, typically there needs to be experience in the empiric manner with one or more therapeutics with one or more mechanisms of action where the result on imaging is correlated with the clinical endpoint.
Great, thanks for that comment. One of the listeners has also asked a question for Dr. Kresge that they've noted that some MRI scanners are already equipped with MRI PDFF capabilities directly on the console, such as the GE Ideal IQ system. So why is it useful to have a central lab managing the imaging data for a clinical trial and calculating PDF maps for central review if it's available on Philips and GE and Siemens consoles?
Mm-hmm. I guess the main reason is to reduce variability that occurs across scanner platforms. With central lab managing the imaging data, we are acquiring the same MRI sequences. We are performing PDFF analysis the same way for all sites. This leads to reduced variability in the assessments and more robust results. And also, we need to keep in mind that by using the central lab, we are in position to combine the benefits of using Rave Imaging for image management and the simplicity of single pipeline for quantitative analysis. So these would be the ones I would quote as being the most important.
Thank you. Dr. Gleiss, there's a question, I think related to your presentation, basically saying that Rave Imaging seems like it makes it really easy for sites to manage their own imaging data and to provide it to a sponsor along with their own RECIST assessments for disease progression. So when do you think it's necessary to utilize the services of central readers or a core lab like MedPace? And what benefit does that bring to a trial?
So if a site's willing to utilize the Rave metadata system for their reading, it can help them be more precise. But even still, it relies on the individual radiologist selecting the lesions. And with central reading, you get theoretically the potential for more repeated uniformity, as well as following the central imaging charter literally to the letter. With multiple sites, there could be concern that it's not followed literally to the letter. With RECIST, that variability is a whole lot less than when you're not in RECIST 1.1. But nonetheless, I believe it's always safer to at least have the film sent into a central imaging facility. If you never know when you're starting the study how close you're going to come into your target p-value. So if you beat your target p-value by a whole lot, it mattered a whole less. But if you only beat your target p-value by a small amount, having the central imaging to demonstrate that it's really clinically meaningful, if that was key to your primary efficacy endpoint, is going to turn out to be key. You're going to get less challenges from the regulators that way. So you never know what the results will be, and therefore it's safer to have things collected prospectively so that they can be read.
Great, thanks a lot. I think that just about wraps up our hour. So thank you to all of the listeners. Thank you to the panelists and our experts for a lot of insights about quantitative imaging biomarkers for clinical trials. And also thank you to Medidata for your participation. So I'm going to turn it back over to Michael to conclude things for us.
Thank you, Scott. And to let our audience know, this webinar has been recorded, and you will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. On behalf of Medidata and MedPace and our panel, thank you for joining us, and we look forward to seeing you at future events.